Hello, this is Mr. Russell, your AP Biology teacher, and today we're going to begin talking about membrane structure and transport of molecules. So the first thing we need to talk about is the cell membrane. We know the cell membrane is selectively permeable. So the cell selects what materials can enter or exit the cells through the membrane. It's kind of like a screen over a window. The screen will let air into or out of a house or a building or a classroom. But it's also going to keep things like big bugs or maybe people or leaves from entering or exiting. So therefore the screen is selectively permeable, just like the cell membrane is. Now if we look at the membrane structure, it's made up of phospholipids, and phospholipids make up the majority of the cell membrane and also organelles membranes as well. So there are a couple things you need to look at here. I want you to underline this term amphipathic molecule. So phospholipids are amphipathic molecules. What that means is, I want you to highlight this, it just means that there is a hydrophilic and a hydrophobic component to it. So these molecules create the bilayer, and the structure is held intact by the presence of water outside and inside the cell. And that's done so by this negatively charged phosphorus that line up to make their barrier, preventing water from forming what's called a hydration shell. And that's just where water surrounds the molecule around the phospholipids, and thereby it ends up dissolving the membrane. That would be a bad thing if our phospholipid bilayer starts to get dissolved, which is what a hydration shell can do. So I want you to underline the terms amphipathic molecule and hydration shell, and I want you to highlight the terms or what their meanings are on both of those. So let's talk a little bit about proteins. These are also amphipathic molecules. So this is due to the protein folding into a 3D structure, and that proteins are composed of amino acids, which some are polar and some are nonpolar. So there's two types of proteins that are going to be present in the membrane. And we look at these. The first one, let's see if I can get this for you. First one we'll look at here are going to be these integral proteins. Here's an integral protein. Here's an integral protein. So when we think about integral proteins, what does that mean? They run completely through the bilayer from the outside to the inside. The function is the transport of molecules and foundation. So it helps to maintain the integrity of the structure. So you can see here that this one right here runs from one side all the way up to the other. Same thing here, and same thing here, and here, and here. So when I think about this, I think about not only does it run all the way through it, but it goes into. So an integral protein runs into or in through the phospholipid bilayer. Well, a peripheral protein, such as this one here and this one here, these are going to be on one side or the other of the phospholipid bilayer. They do not extend into the bilayer of the membrane. So these act as sites for attachment of the cytoskeleton on the inside of the cell, and they act as attachment for the extracellular matrix, or the ECM, on the other side. So if we think about the ECM, it's just kind of like an armor for the fragile cell. So in this case, we're looking at the outside here so that this extracellular matrix can attach to these integral proteins, whereas on the inside you have an attachment for the cytoskeleton that you're going to see here. They're going to be these little orange looking structures. Alright, so when we talk about these, we've got there's two different types of proteins. We said there's going to be integral proteins and peripheral proteins. Well, the proteins of cell membranes, they have several different functions. And we look at these first three here. You've got molecule transport, just helping move food, water, or something across the membrane from one side of it to the other. They also act as enzymes. They have to control the metabolic processes. They speed up and slow down reactions. They also help with cell-to-cell communication and recognition. so that the cell can work together within its own tissues. If we look at a few more of these, we got signal receptors here, or signal recognition. These just act to catch hormones or other molecules circulating in the blood. Another function of these proteins are going to be intercellular junctions. So they're going to kind of stitch together cells to be able to make tissue, make tissues larger so that we can end up with things like organs and organ systems and organisms. They also act as attachment points for the cytoskeleton and the ECM, which we talked about a few minutes ago. Now, we're thinking about the phospholipid bilayer, the cell membrane, the structures. We've got to think about cholesterol. And the cholesterol, this molecule, we said already in our class that it helps to keep the cell membrane flexible to some degree. It also helps to keep the cell membrane of plant cells from freezing solid in very cold temperatures like in the tundra. We also know that too much cholesterol can be bad for us too. We know that there are some health-related issues. But in this case, our bodies and our cells, they know what to do and they know how to keep themselves working properly for the most part. We don't have to do it for them. It can take care of itself. So the membrane, when we think about phospholipid or the phospholipid bilayer, this membrane is described as a fluid mosaic model. I want you to underline that term as well. And the reason is because it looks like a moving or fluid puzzle, which is often referred to as a mosaic. All the pieces can move laterally, kind of like you guys. If you want to move from seat to seat in the classroom, you can do that. So the proteins move in this sea of phospholipids. Be kind of like me or the teacher moving around the classroom or moving around your desk. So if we think about the ceiling and the floor being water molecules, these kind of keep us in place. They kind of keep us from going up too high, or they kind of keep us from going down too low. They are a barrier that keeps us in the room itself. So we're thinking about cell membrane, cells in themselves. We've got to think about the importance of surface area to cell volume ratios or relationships. This is key. You're going to see this over and over and over. This is possibly one of your math questions. It's going to be in there somewhere on your AP exam. All cells are considered to be open systems in their natural settings because there are materials coming into the cell from the surrounding environment, as well as materials leaving the cell and going into the surrounding environment. So the cell is open to interactions within its environment. Remember, cells can only be so small. There has to be enough room or volume to hold things and to perform work inside a cell using the cell membrane. Cells can only be so large. Larger means that more traffic going in both directions across the cell membrane. So a cell must be large enough to contain DNA and ribosomes for making proteins and some cytoplasm to act as a working space. They can only be so big because they have to be able to move that food into and waste out of the cell efficiently. That's key here. If it's too large, the cell becomes inefficient at moving these things, so it divides to get back to a smaller state. How do you increase surface area without increasing the cell volume significantly? Well, you do that by folding membranes. Let's look at the following examples. If you think about your lungs and other respiratory surfaces, the surface of the cell needs to stay wet for gases to exchange within themselves for that to take place. If you notice the increased surface area by folding, this allows for the transport of the massive amounts of oxygen that organisms need to live, as well as CO2 gas, or to be able to get it out as a waste product. So if you look here in these little structures right here, you get lots of folding there within those membranes. Get a large surface area. You increase that surface area, you can increase the volume that's going to be taking place there. So your lungs have surface area that are equal to the size of a tennis court. It's all just kind of wadded up and folded up there inside your chest. If you take your lungs and you unfold all those little folds, about the size of a tennis court. That's pretty impressive. If we think about ourselves and animals and our intestines, well, here again, we look at lots of folding. There's going to be folds here. Lots of folds going on here within the stomach. And then even inside of there, you get other folds there. This is all inside of our intestines. Now, so if we look at the folding at the organ and at the cellular levels, this allows your body to take in massive amounts of food that we eat. After all, you have 1 trillion cells to feed, and they need food all day and all night long. So this section of intestines has these little finger-like projections in them called villi. Oops, sorry here. Let me get to where we're supposed to be. So if we look at this inside of our intestine, we've got these little villi. Circle that word. This just increases surface area. These little small finger-like projections also have these little microvilli in here. So here are these folds. And then you've also got little folds on top of folds. And here you can see it again, looking at these little structures. This increases surface area as well. So these villi and these microvilli, these are located in the cell covering. So this increases surface area. If you take your intestines and you were to unfold all the folds of the villi and the microvilli, well, this total surface area is going to be equal to about the size of a football field. Very, very large. Lots of surface area. Increases workspace. It increases the ability for your cells to be able to be more efficient, be able to transport materials into and out without causing any problems. So, we as humans and other animals, we have excretory systems. So there are thousands of cells that all try to help purify the liquid components of our blood by performing selective molecule transport. The building up this nitrogen containing ammonia, comes from our cells utilizing proteins for energy production. Well, our excretory system gets rid of the ammonia either as straight ammonia, in the case of fish, as urea, as in most land animals, including humans, or as uric acid for birds and reptiles. And also, I want you to notice that urea and uric acid get rid of additional waste product, carbon dioxide. So remember, this is part of the nitrogen cycling. All right, that was it for this part of today's lesson. We will continue tomorrow. Thanks. Have a great night. I hope that this is beneficial and helpful.